All right, thanks everybody so much for coming. Can you hear me in the back well if I'm talking at this? Okay, thank you so much. So I'm really excited to be here today to talk to you about some of my research and plans for future directions. The broad scope of my research is about host-microbe interactions, especially in the Anthropocene. Oh, okay, there we go. So today I'll talk a little bit, I'll introduce myself a bit and talk about kind of the broad overarching questions that I'm seeking to answer and tell you a bit about how I got to them. Then I'll talk about some projects that I've done in the past or are ongoing and how they informed my future directions. I'll go on to talk about a few future projects that are kind of planned for the next 5 years or so. And then in the end, I'll talk about some of the policy engagement that I'm currently working on and hope to do in the future around areas like host microbe interactions, biodiversity conservation, and managing spillovers of zoonotic disease. So during my PhD and prior to my PhD, I worked across a variety of systems, mostly in amphibians and reptiles. And primarily, I was concerned with looking at population genetic patterns, patterns of diversity in microbial communities, and how those two interacted. So I was really looking at quantifying and understanding patterns of diversity in the natural world. And as I turned towards a postdoc, I really wanted to get more into the mechanistic processes that caused those different patterns of host diversity and microbial diversity. So I started with the Cornell Institute of Host-Microbe Interactions and Disease, looking at interactions between reptiles, their immune genes, and their apicomplexan parasites. So these are the lizard malarias and other similar obligate single-celled eukaryotic parasites. And then I took kind of a more integrative turn in my research and started working my second postdoc with the Lancet Commission on the Prevention of Viral Spillover, looking at particularly zoonotic disease spillover to humans and how to prevent that. So Lancet commissions are set up by the medical journal, The Lancet. They bring together experts to kind of take a look at big, broad questions in public health and put forth recommendations that will be implemented in public policy and public health practice for how best to handle these big global challenges. So the specific Lancet commission that I'm with is on the prevention of viral spillover, with a particular attention to preventing future pandemics. This is a collection of 28 experts from across the world. We're equally split between global north and global south, and it's a broad range of expertise, everything from disease ecology, epidemiology, virology, all the way to indigenous knowledge, public health practice, and economics. So it's been really a crash course in how to do interdisciplinary research, a lot of really synthetic thinking. I'm one of 6 or 7 postdocs that's supporting the Lancet Commission. My personal projects are kind of laying down the foundational understanding of what we can and cannot say about the current state of knowledge of zoonotic diseases worldwide. So the 3 major projects that I'm working on are identifying viral traits that predict whether a lineage will have emergent pandemic viruses. How are they the same or different from pathogens, viral lineages that might spill over but won't go on to spark a pandemic? My second major project is defining whether we can use data from one set of viruses with particular ecological characteristics to predict the process of how pandemic viruses might spill over. So the real kind of nuts and bolts of this are we have a rather big data sets on spillovers of rabies, when and where that happens, spillovers of things like hantaviruses or henipaviruses, so Lassa and Nipah viruses that are kind of in the center, their center of diversity is in Southeast Asia. And so these are more data-rich sources, but they don't go on usually to cause big outbreaks or human-to-human transmission events. Whereas things like avian influenzas or COVID definitely do, but we have very little data about the circumstances around their spillovers because usually it's a few isolated spillovers, one or two of which will go on to cause big chains of transmission. So there's just a real data limit there. And so what we're asking is, can we make determinations about the ecological processes that lead to rabies spillover and apply those directly or indirectly to a new COVID spillover or a new influenza spillover? And we want to try to get as data heavy and mechanistic to answer that question as possible. And finally, I'm testing the idea that rates of pathogen spillover are accelerating. So if you look at the raw number of reports of pathogen spillovers, they are going up. But at the same time, our detection capacity is getting so much better, our technology is getting so much better, and internet access is getting so much broader and more accessible throughout the world. It's really hard to tease apart detection and reporting improvements from a real increase in the rate of pathogen spillover. So I'm kind of helping to lay the foundational work for the main question of the commission. The big goal is to reduce the rate at which zoonotic viruses spill over to humans. So to think about this, I want to kind of step through the whole process of a zoonotic spillover. Most of the time, for most viruses, they're just circulating in nature and they're in whatever reservoir host species they have, and there's really very little risk to humans or our domestic animals. But every so often, something will change. Something will happen in the environment that forces the hosts closer to humans, or humans might come closer to the host so that there's now more overlap in space and time. If at the same time as the overlap happens, the hosts are shedding a lot of virus into the environment, then humans or our domestic animals might be at risk of getting infected. If that happens and the virus is capable of transmitting between humans, you might have an outbreak. And if the outbreak spreads more rapidly than the local public health capacity can contain it, you can go on to an epidemic or pandemic. So really a lot of different processes have to happen in order at the same time for a spillover to occur. And the goal of the commission is to just throw up blocks in as many of those places as possible, reduce overlap, reduce shedding, reduce the risk that the first spillover will go on to trigger an outbreak. And as a group, we consider those primary preventative interventions, everything that happens before the first human is infected. And those join better known secondary preventative approaches like vaccines, testing, masking, everything we do to try to contain a virus once it's already in humans. We just want to add an extra layer of prevention and hopefully reduce the total number of pandemic outbreaks by putting more and more blocks in the way. But there are some really big challenges with this. The first one is spillovers, scientifically speaking, are rare. It's really unusual to have somebody sampling in the exact space and time when a spillover happens. So a lot of what we have to understand about spillovers and the way we get at those data are by looking at reports from later, collating those. So really trying to understand the exact mechanisms is very challenging. The second problem is that conditions that drive a spillover can be very fleeting. So the exact set of circumstances that cause a host to overlap with humans and cause them to be shedding more virus than usual might just be a slice of days or weeks. And if you have to wait for an outbreak to happen and to get diagnosed and then stand up a team and get back to that location, then the circumstances might be completely different. So you're no longer sampling the drivers of that initial spillover event. And then finally, a big impediment in terms of convincing governments or any other public health body to implement primary prevention is that it's really, really hard to prove that your investment prevented a pandemic. There's just no real way currently to measure that. So thinking through all these and thinking what I brought to the table, I kind of reframed this in terms of my background in community ecology. And I thought really a spillover from an animal reservoir to a human is just one instance of a host switch. And there's no like fundamental difference in a virus going from a bat to a person. than a virus going from a species of mouse to a species of lizard. And so that helped me really firm up some ideas about how to get at the mechanisms of spillover in a biological community in a way that could be really analytically rigorous and well replicated. So I'll walk you through kind of the schematic I have for how to think about spillovers and trying to get at their drivers. So to reframe in my head, I thought, what would be the characteristics of a community of animals, a biological community that's extremely spillover prone? What's the worst case scenario? So I'll build that community and we can then think back about how to test what causes it and how to kind of interrupt those processes. So first of all, one of the really clear empirical signatures in the literature, one thing we can feel really solid about in terms of understanding spillover is that it's more common when habitat disturbance occurs. So starting from habitat disturbance, thinking about a change in a host and microbial community, you get a lot of turnover in the hosts. And we know that host communities in disturbed habitats look different from host communities in intact habitats. Our understanding of how habitat disturbance hits microbial communities is still developing, but it's pretty reasonable to assume that there's turnover there too. At the same time, what works well in terms of immune strategy, how much energy a host put into their immune defenses might look different in an intact habitat relative to a disturbed habitat. And there's two pieces of empirical data that really inform this and made me want to include it in this schematic. The first is that spillovers happen more often when host immune responses are depressed. And this can be from nutritional stress, other sources of stress, or even just because there's a host that generally invests less in immune defenses. So when the host is not mounting strong immune responses, then pathogen titers can grow greater in that host. And that in turn leads to a greater amount of shedding of the pathogen into the environment. And so there's just more total pathogen there for a human or a bridging host to pick up. You're increasing risk. In addition, from invasion biology, there's the observation that invasive species are often very successful when they reduce their immune investment. And this could be because they're coming into a new area and the local pathogen fauna is not well adapted to infecting them, so they don't need their immune response as much. Or maybe it's an evolutionary strategy where they're accepting a certain amount of pathogen pressure as a life history strategy, but allocating energy more towards reproduction or growth and kind of just accepting that increased pathogen load as, you know, an unavoidable, but ultimately not a deal breaker in terms of their life history strategy. So integrating that, I wanted to really understand how host immune investment, the total energy budget toward immunity, might alter between intact and disturbed habitats. And finally, the sum of these two processes of turnover in the communities and differing host immune strategies could lead to an increase in host switches in the disturbed habitat. So if you have a set of hosts that are immune suppressed, either because they're stressed by disturbance or it's an adaptive strategy to invest less in immune defenses, new microbes might be more successful in starting to infect that suite of hosts. And so all of a sudden, you have this new adaptive space for all the microbes to kind of work through. And they have this opportunity to start adapting to a novel host and increasing their rates of host switching. And of course, this is a really key component of the process if we're thinking about pandemics, because one further empirical observation about viral lineages that go on to cause human epidemics is that they make big jumps in their host phylogeny. So for example, you can think about hantaviruses, which spill over pretty frequently, but they don't really go on to cause epidemics. And although they do infect humans, they really just kind of focus on rodents. That's their main reservoir host. So they might be making jumps within rodent host phylogeny, but they're not making huge jumps. And you contrast that to something like avian influenza, which will jump from a bird all the way to a human or to a cow, these big phylogenetic host jumps. So that's a kind of repeatable, consistent empirical pattern in the pandemic pathogens that we're really concerned about. So this could encourage the ability for lineages to make these big host jumps and start adapting to that different life history strategy across the host phylogeny, which of course leads to spillover, the ultimate process we want to prevent. And finally, over all of this, there is the aspect of sampling bias. Can we adequately sample in the field all of these hosts and microbes? And when we're starting to think globally, can we account for the biases in detections and reporting pathogens across space and time? So my ultimate goal is to integrate across all of these processes, understand them in one single system or set of systems, and be able to make strong predictions about factors that increase or decrease risk of pathogen spillover from a specific community undergoing particular disturbances. To make it a little more concrete, I boiled these down to testable questions that I can get started on immediately. One of my main questions is, what is the effect of habitat disturbance on host immune strategy? If we're sampling populations of animals in intact versus disturbed habitat, can we make consistent predictions about how much energy they're going to be putting in immune defenses? What factors, ecological or morphological, physiological, predict if a microbe is going to A, survive through a habitat disturbance, and B, start infecting new host species in the disturbed habitat? Can we make consistent predictions about which microbes might follow this life history strategy that could select them towards higher emergence risk? And finally, what are the best sampling strategies to understand all of these processes in my own data set, in my own practice, and also when looking to integrate broad data sets of pathogen spillover? So prior to now and in the future, I have used reptiles as a model taxon, and I firmly believe that they are one of the best vertebrates to study evolution because they do all sorts of wild things with their life history strategies. First of all, even between lineages and even within a species, there's dramatic variation in how much energy different populations of reptiles put towards immune defenses. So they're a great system to study these questions in. Second, they have some of the absolute classic adaptive radiations in nature. You know, one of the classic examples are the anole lizards on the Caribbean islands, where they've radiated into different morphs repeatedly. There's also repeated evolution of really interesting physiological processes in reptiles multiple times, so you have this statistical robustness. One example is in the repeated evolution of live birth. Evolution of live birth happened once in the mammals, 0 times in birds, and more than 60 times in snakes and lizards. And you also have repeated evolution of parthenogenesis, where some lineages of snakes and lizards in nature will become all female and start reproducing clonally. And those are just a few of the examples of why snakes and lizards are a great model taxon for evolution. And now that we have sequencing technology where it's much easier to generate a robust reference genome, it's going to become much more tractable to really use them to their full potential as a model system. However, I did say I was really interested in the big phylogenetic jumps. So to accommodate that, I do plan to start adding in mammal sampling, bird sampling, and more amphibian sampling to get the biggest possible profile of a biological community and really capture those big host jumps. So I've started working a bit with bats, and I'm really looking to add more diversity into my sampling strategy. I also sample across microbial phylogeny as well. I worked with bacteria, viruses now, and pathogenic eukaryotes. All of these have emerged as novel pathogens in humans, and they also all interact in an ecological system. So the broader diversity of microbes I can capture the less likely there is some X factor that I'm just missing that's biasing all my results. So I do try to go as broad as possible with the microbes as well. So now I'd like to go through a couple of my previous and ongoing projects that have informed the questions I'm asking. And the first one goes back to the question of how immune investment changes throughout invasion biology. And for this, I use the brown anole, which has been extremely successful in establishing new populations throughout the southern US. For this question, I looked at the major histocompatibility complex, which is an immune gene, which functions in pathogen recognition. So it's the one that notices there's an invader and sets off the whole rest of the immune cascade. And MHC is a great system for understanding local selective pressure for a couple of reasons. One is that it has to match whatever non-self parasite or pathogen simply by chance. And so it functions like a lock and key mechanism. You can see here it's a membrane-bound protein that interacts with a peptide and then sets off the signaling cascade. So in order to mount as robust an immune defense as possible, this gene has been duplicated in vertebrate genomes many times. So each vertebrate has a suite of immune genes, a suite of MHC genes. And for these MHC genes to function, they also mutate very rapidly. So there's a bunch of different copies, and each copy between individuals within a population is mutating quickly. When pathogen pressure is high, you see repeated selective sweeps at individual immune genes. So there is a rare allele advantage because the local pathogen population is going to be evolving away from being recognized by any of the existing immune gene MHC alleles in the population. So novelty is a huge benefit for whatever animal is carrying this new MHC variant. So with that in mind, we expect a lot of turnover where parasite selection and pathogen selection is high on a population. Going back to the history of brown anoles throughout the United States, we have this really great temporal transect of how recently different populations were established. So brown anoles are native to the Bahamas and Cuba. I wasn't able to get any Cuban samples, but I do have some samples from the Bahamas in the native range. About 150 years ago, they made the hop over to South Florida, and then they moved up the panhandle successively over the years and into Louisiana and ultimately into Texas. And that's kind of the scope of the more natural expansion of brown anoles in the US. In addition to that southeastern US distribution, there have been a couple of independent introductions of anoles to Southern California, the LA area, and to Hawaii, mostly in nursery plants that come from Miami. So they're great at like burrowing down into those plants. They get shipped across the US, and all of a sudden you have a new population of anoles. So for each of these points on the map, I sequenced a suite of MHC1 genes in each individual. And then because MHC1 ultimately descends from an original ancestral copy, you can put those different MHC alleles in a phylogeny. So I looked at the phylogenies here, and each of the tips represents a different MHC allele in the population. So a single host will have multiple points on this phylogeny, but overall, this is the full suite of immune genotypes that a parasite or pathogen would experience if it was trying to infect this population of lizards. And here you can see some evidence of selection because this phylogeny has the classic shape of a phylogeny that's been experiencing repeated successive sweeps. That is, all of these tips are quite close together, and you have these longer internal branches. So everything's turning over quickly in this phylogeny. It's evidence of selection in the specific way that MHC experiences selection. And this is an MHC phylogeny from a population of South Florida individuals. So they're an older population. The local microbes have had longer to adapt to them, and they're also physically really close to their native range. In contrast, here is an MHC phylogeny from Hawaii, where they're quite far from their native range. And you can see in terms of the shape of this tree, much longer branches at the tips. And this is what we expect from a more randomly evolving set of trees, a real set of genes, a real big distribution of shorter branches, longer branches. And this is a tree that's experiencing less selection. Here's a nice little comparison just of these little clades that happen to be at the bottom of these trees. Long internal branch, short tips, and much longer tips down here at the bottom. So by quantifying across this whole gradient, I can determine when you start to see a switch in terms of how long a population has been established from a weaker selective set of selective pressures to a stronger set of selective pressures. In the future, I'd like to apply this kind of process, looking at immune genes and the selection pressure on them, to broader host populations so I can get back into that community ecology context. Moving on to some work I've done with bacterial microbiomes that include commensals, mutualists, and pathogens. I wanted to determine what was structuring the diversity of microbes across different host communities. And so I sampled along a gradient from Michigan all the way down to Peru. And along that gradient, the diversity of the host community increases tenfold, from about 20 total snakes in Michigan to over 200 snake and lizard species that occur in the Amazon basin in Peru. And across that whole gradient, I wanted to test the hypothesis that local host diversity would increase the total diversity of microbes available in the environment. So I used a metabarcoding approach, looked at the 16S rRNA in cloacal microbiomes in lizards and snakes across this gradient. And you can see that the point shapes are scaled by the number of species I was able to sample in each location. So looking across the gradient, bacteria diversity did not, in fact, increase. It was remarkably stable across all populations. And this is both within hosts and between hosts. So there are clearly different processes structuring microbial communities compared to host communities. But digging a little deeper into the data, I was able to pick up some differences. So what I found is that bacteria in our higher host diversity tropical communities tend to be much more host specific. This is a measure of phylogenetic specificity of hosts for each microbe. So microbes that are infecting hosts that are really phylogenetically similar are going to be in the below zero parts of this y-axis. Microbes that are much more phylogenetically host generalists are going to be in the higher areas above the zero line along the y-axis. And you can see, it's always a messy trend with microbiomes, but you do see a real shift towards communities that are more dominated by more host-specific microbial lineages. To get even a bit deeper into the process, I wanted to look at the ways bacterial lineages co-occurred or kind of repelled each other across hosts in these communities. So here is an example of bacteria lineages that might co-occur or both be together in hosts more often than you would expect by chance. If you look at the blue and yellow microbe lineages, you can see that they're always together across many different host species, whereas this green lineage is much more host specific to the geckos. And so for every pair of microbes in my different communities, I counted up the number of times that they were always occurring together across my hosts and compared those to what we would expect by chance. So for one location, this yellow box plot are the number of pairs that are co-occurring across many different samples of a host microbe matrix. And then if I randomize the matrix, I get far, far fewer pairs that are co-occurring in the randomized matrix. So there's a non-random process where bacteria lineages are associating. If I look at places where the pairs are anti-co-occurring, like the yellow and blue are never occurring in the same host, there's no difference compared to what I would get by chance. So there's a strong bias towards positive co-occurrence in these data sets. And looking across all of my locations, there's an even higher frequency of these co-occurring pairs in the tropical systems, where we also see this shift towards a lot more host specialization. And the reason this really piqued my interest is that the lineages that tend to be in these co-occurring pairs come from bacteria taxa that are opportunistic pathogens on humans. So things like Salmonella, Shigella, the things that we hear causing outbreaks of foodborne illness are showing this pattern. For future directions, I'm interested in seeing how habitat disturbance might play into these processes. So when we have a habitat disturbance, are we filtering out these specialist bacteria and leaving only the generalists that might be more in this kind of opportunistically pathogenic group? If you lose the specialists from a community due to habitat disturbance, does that create more ecological opportunity for the generalists? And finally, if we do disturb a community, what happens to these co-occurring groups of bacteria? Do they get stronger? Does that fall apart? These are all really interesting areas that I'd like to pursue in the future. So going back to the overall schematic, with some of that background in mind, I wanted to talk about how I plan to integrate all these processes across systems and start testing this idea of what's the worst case spillover community. For all of my locations, all of my potential study areas, I have a set of kind of consistent methods that I would apply just to get myself empirical data. The first would be to collect as broadly as possible. Use 16S rRNA profiles to look for tissue-associated microbiota. So this could be gut, blood, kind of respiratory, any tissue system you are particularly interested in. And also collect data on correlates of host immune investment. So things like white blood cell count, relative spleen size, response to lipopolysaccharide challenge, which is kind of taking bacteria, kind of the shell of a bacteria, injecting it and seeing if the hosts mount an immune response to that, or looking at using transcriptomics to look at relative immune gene expression profiles. With these kind of broad data collected across the community, I can then select some target host species for whole genome sequencing and whole metagenome sequencing to really understand the exact interplay of both microbial and host genetics in a more restricted number of lineages. And then finally, if I can develop hypotheses from those genomic and metagenomic interactions, I can develop some high throughput, low cost screens for pathogens of interest and immune genes of interest, or other genes if something interesting pops out there. And this would allow me to expand back to the broader host community. And what I like about this structure is that each of these steps builds on each other, but they're all independent. So these can become projects for students from undergraduate, master's, or PhD quite easily, and be their own unique unit that's a real contribution and a complete project. So I'll talk about some specific systems that I'm excited to apply this in. The first one is looking at microbial and host diversity and interactions across altitudinal transects, so capturing temperature variation and also disturbance gradients in Central California. This is part of an ongoing broader project that I'm part of, which is called the San Joaquin Desert Comparative Genomics Project. And this is through the Bureau of Land Management, USGS, and the California Academy of Sciences. So the basic layout of the broader project is that we're taking local lineages in this really interesting area of Central California, the Transverse Ranges, where you have the Mojave meeting the Central Valley, and there's just a lot of biogeographic complexity in that area. And we're going to generate or have generated high quality reference genomes for these species. And then also use low coverage whole genome sequencing to do really detailed phylogeography of the way different lineages within each species are arrayed across this complicated landscape. So these are the first 4 of the focal species. And within these focal species, we have both habitat specialists and generalists, and animals that are locally common and locally rare. So we're really capturing a lot of ecological diversity of reptiles in this area. It did start with reptiles, but the plan is eventually to expand to mammals, arthropods, and plants as well. So a real full community approach. My part of the project has been on Xanthusia vigilis, the desert night lizard. And this is from an earlier sequencing project I did with this group. We looked across a lot of Central California. We sequenced populations further north in the Coast Ranges, right here in the core area of the Transverse Ranges and in the Mojave Desert. And in this project, we found this really interesting contact zone right here between lineages that are more associated with the Mojave and more associated with the further north populations meeting in this really small area in one specific part of the Transverse Ranges. So that was exciting in terms of phylogeography, but also opened up a lot of really interesting questions about how we can use these kind of replicate evolutionarily divergent lineages in the really similar habitat to test questions about host and pathogen interaction in the context of disturbance, but in a way that's essentially evolutionarily replicated because these 2 lineages are really not exchanging genes whatsoever. So in this context, what I'd like to ask is, what can we say about selection pressures on immune genes that we can quantify in the 2 lineages? This is work in progress. We are about to publish the genome paper for Xanthusia vigilis, and we have low coverage whole genome sequencing across the contact zone in the Cuyama Valley. So we'll be able to quantify selection on immune genes as part of that project. I'd also like to look at alleles in the genome that are associated with tolerance to habitat disruption. So the populations we've sampled have experienced different amounts of habitat disturbance and also are associated with the thermal niche and ability to withstand desiccation in these animals. This is an area that could apply to climate change research as well, and the interaction between thermal stress, water stress, and pathogen tolerance. And then finally, I would profile the bacterial and viral metagenome and associate how the presence of different microbial, potentially pathogenic lineages are associating with the alleles we focused on in the first 2 aims. And so this is really a host-to-microbe type project that already has some preliminary data. Moving on to a more microbe-first project, this is a set of ideas that I could apply to a lot of different locations, but I was looking on iNaturalist, as you do, and I noticed right here around Carbondale, you have a really great example of how different snake lineages are more or less tolerant to habitat disruption. So the idea would be to put sample sites on a transect from more intact to more disturbed habitat, sample along those transects. So you have great examples like the rat snake and garter snake, which seem to occur in all sorts of places. They don't mind if they're in town or more in the forest. whereas things like the green snake and the brown snake are more restricted to the more intact habitats on the edge of town. So taking a disturbance gradient like this, if you're sequencing microbes, you can start asking questions like, do we see a loss of specialists in more disturbed habitat? And so here is a generalist host. It has its bacterial, its suite of associated lineages, more specialist hosts with its specialist microbes in intact habitat. If we experience disturbance, are we going to lose these specialist microbial lineages, even if we still have the specialist host species that's clinging on after the disturbance? And if that happens, are some of the microbes associated with the more generalist hosts going to jump in, have that host switch to this newly open area of opportunity for them in the disturbed habitat? So to make that a little bit more concrete, I, again, broke it down into 3 specific aims. The first would be to sample across these disturbance gradients and look at correlates of host immune investment along the disturbance gradients. Are they increasing or lessening with greater degrees of disturbance? And is that consistent across host species? Then I'd like to quantify whether we see that jump in host breadth, whether microbial lineages are expanding their phylogenetic host breadth in more disturbed environments, and can we pick up any signature in their genomes of what selective processes the microbial lineages are experiencing as they jump to a novel host. And finally, I'd like to determine if hosts that invest less in immune defenses are more likely to receive new microbial lineages or to be donors of new microbial lineages to other hosts in the community. Thinking back to that idea that hosts that invest less in immune defenses are more likely to be a source of spillovers, where immune suppression causes, for example, a pathogen to spill from bats into humans. Do we see that in this broader context? So the end goal, of course, is to integrate across all of these processes, really understand how that's happening across full communities. But I think those are some tractable places to start that I'd be really excited to work on. So finally, I'd like to talk a little bit about my work in policy, how we actually take these ideas that I'd like to develop and turn them into real-world impacts. So right now, my primary work in the policy space is working with the World Bank Pandemic Fund through the Lancet Commission. The World Bank Pandemic Fund is a cooperation between the World Bank and the World Health Organization. It was stood up 2 years ago. It has 1 more year to go officially. And the goal is to provide donations to lower and middle income countries to increase their capacity to detect and respond to spillovers. Ultimately, the Pandemic Fund hopes to leverage $3 billion in direct donations and matching funds from from national governments and non-governmental agencies. So our part of it is to encourage more data collection on the ecological processes that lead to spillover. And this gets back to how I was talking earlier about the big challenges in primary prevention. So the heads of the Lancet Commission went to the World Bank Pandemic Fund and said, can you offer grants to offer calls for proposals for more ecological environmental surveillance? And they said, that sounds great, but how do you do it? And that's a very, very reasonable question because there is no guidance. So with the Lancet Commission, I'm working on developing that guidance, surveying different processes or different approaches that have worked in the past on collecting data to really understand the whole process of pathogen spillover and presenting that in a way that can guide calls for proposals and ultimately proposals to collect these data as part of routine pathogen surveillance. And ultimately, what we hope is that we can create a robust data set, not just of when and where pathogen spillover is happening, but also what was happening in the broader environmental context when those spillovers were occurring. And that can ultimately inspire new primary prevention approaches. So really creating the empirical database to create solid evidence-driven primary prevention approaches. Here too, I have a basic set of steps that I apply to all sorts of questions within the policy space as it relates to zoonotic disease. And this is a very interdisciplinary set of processes. So I work with experts in public policy, public health, virology. It's a big team that approaches any of these questions. But essentially, we try to map out all of the different public policies and agencies that whose purview impacts pathogen detection, whether or not a test gets performed, and whether or not the results get reported. Then we quantify across space and time how those detection biases impact the likelihood that a case of spillover that occurs actually gets reported and gets to the point where we can scrape the data and have that accessible. After correcting for reporting bias, then we look for areas that are at high risk of spillover. And finally, we go back to the public policy and quantify whether we can see whether if a public policy is making people safer or less safe. So some examples of this process, we've started in Florida with West Nile virus. Florida is super interesting in terms of mosquito-borne disease because mosquito management is very local. There's basically 3 ways that mosquito management is done. One is by a county government. One is there's no local government and it's basically managed by the state. And one is a special district. And special districts are interesting because they can raise taxes. And that means that they are far, far better resourced than county governments or places that are managed by the state. And so we have this patchwork here of different levels of investment in mosquito control across Florida. So going forward, we're looking at reports of West Nile virus across Florida and seeing if we can detect biases in terms of detection, reporting, and management in those data sets as it relates to these local public policies. In the future, I'd like to expand the West Nile virus work across the rest of its range in the US. It is a big concern in New York. We had some outbreak years of mosquito-borne diseases in the Northeast this summer. And I know that there are West Nile virus cases here in Illinois as well. I'm also starting to work with a group who is interested in highly pathogenic avian influenza and how it's jumped into cattle, for example. And so we're really interested in quantifying the testing and reporting policies, how those interact with the data we have available, and then how we can understand how those the testing and reporting impacts the evolutionary inferences that we're able to draw. So phylogeny of highly pathogenic avian influenza lineages could tell us a lot about when and how the virus jumped into cows from birds, but only if we're pretty sure we're actually catching all of the lineages that arose from that initial spillover attempt, because otherwise our inference the predictions that come from them, and then how the public policy might interact with all of that. And finally, after the Lancet Commission wraps up, there's still plans to continue engaging with these big multilateral bodies that interact with biodiversity and pathogens, and also climate change. So for example, engaging with the UN Climate Change Conference, the COPs, the G20, the Group of 20 Nations, World Health Organization, the IUCN's Global Biodiversity Framework. So all of these could have a role in really supercharging primary prevention and thinking about how land use can reduce our risk of pandemics. And so that's a process that I would love to stay engaged in the future. With that, I would like to thank many mentors through the years, lots of collaborators, and my really wonderful undergraduates and graduate students that I've mentored and who really made a lot of this work happen. And I'd like to ask for any questions. Thank you so much.